Hi everyone. Today I'll be giving a brief talk about my project. More specifically, I'll be talking about the web application produced for my project named LaminaRGeneVis, the tool to visualize gene expression across the layers of the human neocortex. So to give a little bit of background, the human neocortex is a highly structured part of the human brain with 6 distinct layers. These layers have been determined through efforts dating back to as early as the early 20th century. Since then, the advances in sequencing and imaging techniques have allowed us to determine some characteristics of the individual layers, such as their thickness and unique neuronal populations. However, we've yet to fully understand the cellular, molecular, or laminar architecture of the human neocortex. If we can improve upon this knowledge, we can provide some deeper insight into the layers of the human neocortex, such as layer-specific enrichment of genes that are associated with neurological disorders. While part of a larger thesis project, LaminaRGeneVis was specifically created to be able to examine layer-specific gene expression in the neocortex using existing datasets created from the human neocortex. To gather those datasets, we performed a literature review to collect datasets with 4 main inclusion criteria in mind. Those being that it had to assay the human brain, that it must contain neurotypical donors, that it must contain gene expression data, and finally, that it must contain laminar data. We discovered that there were only 3 such datasets that matched our criteria. The dataset from a publication by He et al. In 2017, data from a publication by Maynard et al. In 2021, and data from the Allen Institute of Brain Science, specifically their multiple region SmartSeq data from their cell types database. For the purposes of time, I won't be detailing these datasets, but here is a summary table of the datasets that we've used. As you can see, there are a variety of sampling techniques and gene expression assays that have been used. So to be able to use these different datasets in our application, we first had to develop a data processing pipeline. This figure here provides an outline of that pipeline implemented in R. We first download the raw data and then process them such that the data were aggregated to the level of each individual cortical layer. Once they were aggregated, we normalized the expression values, first through counts per million normalization and taking the log of those values. This results in the data being represented in a gene expression matrix in the values of log CPM. One note here is that we separated out the Allen-specific data by each major cell type label that they provided, those being GABAergic, glutamatergic, and non-neuronal cells. Now that the data were standardized in these matrices, we could start to build our web application. Using the R Shiny framework and R once more, we used the data as our database and constructed an easy-to-use user interface for users to examine their gene or genes of choice. So the question is, what can we actually do with LaminaRGeneVis? Well, first, you can query the expression of either a single gene or multiple genes, and then compare that expression across the layers of the human neocortex and pre-existing gene expression datasets that we used for the application. It's available at this link down below. And let me do a really quick demo for you in a slideshow format. So once you arrive at the application, you're met with a home page where on your left is the selection interface, and on the right here is what summarizes the data and workflow that's used to create the application. The right will also show you figures that the app generates. Focusing on the selection interface, there are 2 main portions: the mode selection, for which we selected the single gene mode here, and gene selection, where you can type in your gene of interest and select it. Once we press submit, the application will start its analyses and show us a figure with some additional information. By default, Reelin is chosen, so let's go ahead and press submit. Once we do that, the application provides this bar plot as the main figure, where it displays the expression of Reelin across 6 cortical layers in white matter on the x-axis and its expression value as log CPM on the y-axis. Each of these data sets are displayed in their own color, and from this figure, we can see that Reelin, perhaps as expected, is most highly expressed in layers 1 and 2, with it being specifically high in GABAergic cells. But if we wanted to examine multiple genes instead, we could easily select the multiple gene mode on the sidebar. So for multiple gene mode, these 4 genes are selected by default, but you could imagine that you could enter any list of genes, like perhaps a list of genes that are associated to schizophrenia. So once we press submit, the application will again generate figures, but I'd like to focus on the main summary figure, which is the AUC heat map. This heat map in particular tells you what the genes inputted are likely to be enriched or depleted within a given layer of a data set. For this time, the data sets are displayed on the x-axis, and the expression, or rather its enrichment, is displayed on the y-axis. If the AUC value is over 0.5, then that would indicate enrichment of those genes, and values under 0.5 indicate depletion. If a particular value was significant, then a star will be showing within that cell within the heat map to indicate statistical significance. In our case, there doesn't seem to be any significant patterns, but we can perhaps infer that these genes are more highly enriched in the upper layers from layers 1 through 4 in the bulk tissue data. So LaminaRGeneVis here, as demonstrated, can be beneficial to other researchers in its capability to display layer-specific gene expression in the human brain across multiple data sets. Currently, while there are tools to visualize gene expression in the human brain, like tools from the Allen Institute, there are no tools that visualize this kind of expression across multiple data sets. Ours would be, to our knowledge, the first to do so. And as I alluded to just a little bit earlier, researchers could also examine the expression of genes that are associated to neuropsychiatric disorders. In the literature, there is evidence of layer-specific neuropathology in the brains of patients with disorders such as schizophrenia and bipolar disorder. However, the neural underpinnings of such pathology are still not really well understood. Our tool could help by providing an easy-to-use exploratory platform to assess perhaps a layer-specific pattern of expression. With respect to our contribution to open neuroscience, all the code used to create and develop the application is openly available on GitHub at this link. Furthermore, our project uses entirely openly available data and reuses them to promote the reusability of such data sets so that the scientific community as a whole can derive more insights and applications than demonstrated in the initial publications. To finish off, I'd like to acknowledge all of the help to make this project come to fruition, including the professors and lab members who helped with the analyses, and of course, the CONP for the Student Scholar Award that made this project possible. And thank you.